Let me just do a quick introduction. We're not doing full introductions because we have everyone's bio online, which you can get to. These are very distinguished and accomplished people, so it would take a long time. Let me just say that before us here are Professor David Reich. David Reich is an old friend of the Institute, and personally, he's a professor of genetics at Harvard Medical School, professor of human evolutionary biology. He studies DNA and its applications to archaeology, public health, and to our understanding of human adaptation. Dr. Vijay Chandru, there you are. Hi, Chandru. Dr. Vijay Chandru is also an old friend of the institution. He's a visiting scientist in the Global Health and Population Department at the Harvard T.H. Chan School of Public Health. His foundation, Strand Life Sciences, has pioneered the advancement of genomics-based precision medicine in India. And we also have Dr. Priya Moorjani. Priya Moorjani is an assistant professor in the Department of Molecular and Cell Biology at UC Berkeley. Her research focuses on statistical and computational approaches to analyze human genetics and evolutionary biology. And Priya, we hope you will become a longtime friend of the institution. Welcome.
Hi. So I was asked to chair the session. So that's I'm very happy to do that. And we're each going to speak for maybe 10 or 15 minutes, and then hopefully we'll have time for as much time for discussion and conversation as we will for presentations. I think that Priya Moorjani and I have worked together on a number of projects, and so we decided to split our presentations into one chunk, which is what I'll present, and a second chunk, which she'll present. Some of this work that I'm going to be talking about is done with Priya, and subsequent work that she'll be talking about is work that she's done separately from me. And then Vijay will be making a complementary presentation. So I've been intensely working since 2007 on South Asian population history from genetic data on a whole series of papers that have been probably the single largest focus of my group's research program since that time. In 2009, we published our first paper on this topic, which was a collaboration with K. Thangaraj and Lalji Singh at the Center for Cellular and Molecular Biology in Hyderabad. And at the time, it was becoming possible for the first time to collect data from the whole genomes of people using technology. It became possible for the first time to study people at hundreds of thousands of locations in their genetic material. That's routine today, but it was really only becoming possible for the first time just around then. And we were interested in trying to understand how people in South Asia vary amongst themselves and how they relate to people elsewhere in the world. And so we applied this type of data analysis to data we were able to collect in collaboration with these colleagues in Hyderabad. So what we did is we worked with them to collect this type of data from almost a couple of hundred people from 25 very diverse groups that Thangaraj and Lalji chose to be very informative about diverse strands of Indian population history. And so what you're seeing here is the result of what's called a principal component analysis. So what you do here is you take your 600,000 locations in the DNA that people vary. And so it's like a table with 600,000 rows and has maybe 200 columns corresponding to all those dots. Each is a person. And in the entries of the table, there's a 0, 1, or 2, depending on whether a person has 2 copies of one of the 4 letters of DNA, or like 2 adenines, an adenine and a thymine, or 2 thymines, or something like that. And then you multiply this table by itself to see how closely related each person is to everybody else. And you most efficiently separate the individuals from each other based on how closely they're related to each other. And this is a plot of that. The first axis is the most efficient way of separating people. And the second axis is the second most efficient way of separating people. And what you see when you co-analyze South Asian people with other reference populations. Here is Europeans and Middle Easterners, and they form a group of closely related relative to South Asians people over here. Here's East Asian people, as well as some people in northeastern India who are East Asian related, and most people in South Asia are over here. And what we immediately noticed was a gradient of variation with different levels of proximity to Europeans and Middle Easterners and Central Asians, which was very interesting, and we called it the Indian cline. There's also other interesting phenomena, such as people who speak Austroasiatic languages, or who we think mixed in the last thousands of years with people who today are related to people who speak Austroasiatic languages. But we focused on this Indian cline, which contains the great majority of people who live in India and Pakistan today, and comprises almost exclusively or largely people who speak Indo-European and Dravidian languages. And so we had an idea that this gradient that looked like a line might be the result of a mixture between two groups, what we called ancestral North Indian and ancestral South Indian, and that the reason there's a gradient might be that people have mixtures in different proportions of these two source populations, and they mixed at some point in the past. So that was our hypothesis that we proceeded with to find very strong evidence for this particular hypothesis as explaining many of the features, but not all the features of the data. If you estimate the proportion of ancestral North Indian ancestry, the one that's more closely related to Central Asians or Iranians or Middle Easterners, people vary hugely in this estimate, what we call ANI ancestry. And there tends to be people who speak Indo-European languages, more characteristic of the North, with relatively more Iranian and Central Asian relatedness, and people who speak Dravidian ancestry have relatively less, but everybody has, in this estimate, somewhere between 20 to 80% of both of them. Everybody is mixed profoundly between these two ancestral populations. And there's also a correlation to traditional social status in the caste system. And so these are independent of each other, and it suggests that both traditional social status and language have some kind of connection to the proportion to this mixture process that happened in the past. So we were interested in trying to learn about the history of population mixture between this theoretical ancestral populations that we called the ancestral North Indians and ancestral South Indians. And we had three kind of cartoonish explanations for what might be happening. So after the last ice age, the glacial maximum ended 14,000 years ago. Of course, there wasn't ice sheets in South Asia, but the climate and the ecology was very different. And maybe those ecological transitions were associated with movements of people that might have caused these mixtures. Another was that maybe this is associated with the arrival of food production in this part of the world, which is archaeologically estimated in different places to have happened sometime between 9,000 to 5,000 years ago, with different types of indigenous as well as food production, as well as food production related to what was developed in the Middle East and in East Asia, arriving in the region at this time. And maybe it was related to the collapse of the Indus Valley civilization and rise of archaeological cultures on the Gangetic Plain after 4,000 years ago, attested by archaeology. Those were our three cartoonish explanations for what explained the data. So to analyze this, we developed a technique to estimate the dates of mixture, which Priya was centrally involved in at the time, which was published in this paper, Moorjani et al. 2013. And the idea is to estimate when the mixture occurred based on the size scale of segments of ANI and ASI DNA in the ancestral population. And I'll walk you through that idea. So in this cartoon, this is supposed to represent in two chromosomes. You know, you have 23 pairs of chromosomes, chromosome 1, 2, 3, 4, 5, et cetera. So this might be two copies of your chromosome 3. And yellow might be ANI ancestry, ancestral North Indian. Red might be ancestral South Indian. And when these groups are mixing, they might have a first-generation kid who has one parent is entirely ANI and one parent entirely ASI. And so you can think about the great-great-grandparents of someone that you're analyzing, and most of their ancestors might be ANI, some might be all ASI, some people might have one ANI ASI parent, and when you produce a kid, you make a sperm or an egg, and you splice together the DNA you get from your mom or dad 1 or 2 times per chromosome per generation in what are known as recombinations. That's a splice. And so you see these spliced chromosomes appear 1 or 2 times, splice, 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 splice. And then today you have a chopped up genome. And by looking at the sizes of those chops, it's kind of like a dicing of a salad, you can see how long ago it's been since these groups came into contact. It's a very simple idea. And you can look at that size scale and estimate how long it's occurred. You apply this to South Asians, and the answer is 4,000 to 2,000 years. This mixing happens. That's after the decline of the Indus Valley civilization and the movement of people to the Gangetic Plain. So it points to that as the event that brought together these two highly differentiated populations. So another thing that has developed really since about 2015, it's begun possible to do this on a large scale, has been to extract DNA out of ancient human remains. There is really almost no such data in India right now, which is a huge problem for looking at the history of Indian populations. There's been such profound insights into, for example, European and Native American population history, and also now East Asian population history, and also now African population history, based on DNA extracted from ancient human remains. And actually, South Asia is one of the least well-studied places in the world from this perspective. Nevertheless, here's the results of a study from 2019, where we studied not data from South Asia. We only had one marginal sample from studies in that year, but mostly from Central Asia. And we compared it to diverse modern populations shown in dots in South Asia. And what we did is, in particular, we had data from the Central Asian group civilizations that were contemporary to that of the Indus Valley civilization. For example, this is an image of the archaeological site of Gonor, which is from the Bactria-Margiana archaeological complex, which was discovered by Soviet archaeologists in the 1970s and was contemporary to the Indus Valley civilization. They built these impressive, really, cities or towns in places like Uzbekistan and Turkmenistan. And we got data from people, and most of the people are in a main cluster in one of these analyses, like similar to the ones I showed you about before, but there are outliers. And these are outliers who are similar to what you see if you look in a city like Tokyo or something, and most people will be genetically similar. They'll all be in one cluster, but you see occasional outliers, and they're telling you who the people there are interacting with. And so that was sort of, these outliers are super interesting because they're telling you the interactions of these people. So the genetic main cluster are related to contemporary people from Iran, who we also have data from. But the outliers prior to 4,000 years ago are similar to Central Asian and Siberian hunter-gatherers. But after 4,000 years ago, they're related to the pastoralists who swept across the steppe north of the Black and Caspian Seas into Europe, and they also swept through Central Asia, and you see them entering this region after that time. And we also see some people who are mixed with South Asian ancestry at these sites in Gonor. We know from archaeology that these people were interacting with the civilizations to the south, and that there's trade and so on. So we think we may have actually sampled some of those immigrants or traders in this town. So our updated model is not one of two mixtures of ANI and ASI, but three sources. So one of them is a source from the north, from the steppe, which is the least important source, but still a significant one. One is an Iranian-related source, which is just sort of an ancestry type. It doesn't necessarily come from the Iranian plateau. And another is from a peninsular India source, which is related to indigenous groups in South Asia and elsewhere, such as people from the Andaman Islands, but also elsewhere. It's not necessarily from there. And that at the time that we sample these individuals who are outliers, they're really mixtures without any steppe ancestry between Iranian-related and peninsular Indian-related people with a gradient of ancestry. That's the gradient that exists more than 4,000 years ago. And then steppe ancestry pushes into the region, and people have different proportions of that. Then at the decline of the Indus Valley civilization, you get a coalescence of ancestry at this point, and at this point, as people here mix with people here mix with people there, and mixture of these two mixed populations form this primary gradient, which is the one that we see today in South Asia, the ANI-ASI mix. So this mixture of two groups is a collapse of a mixture of three earlier groups who are surely collapses of earlier groups that existed prior to that. This is nothing unusual. When we study other parts of the world, we see layers and layers of mixture at different time depths going back far, far, far back. So I want to switch before moving to Priya's talk to a feature of population structure in India that we already immediately noticed in this 2009 paper, which is that if you take the average pair of groups in India that we had collected data from and measure how different they are based on the squared frequency differences between pairs of populations, we were really surprised at how large this number was. It was much larger than it was amongst European populations. Even after controlling for traditional social status or language group or the state that you were in, it was still much larger than in Europe. And so what this indicated was much more residual population structure in South Asia than there was in sort of another subcontinent of Eurasia, Europe. And so this was very interesting. And what became increasingly clear, and of course many people already knew this, but we didn't know this, was that the truth is that India is not a large population. It's in fact thousands of very many small populations, or small and larger populations. And an example of this is the Vaisya, which I probably mispronounced, from Andhra Pradesh, or now Telangana, 2,000 to 3,000 years ago, what we can actually tell is that this group formed by mixture of the ANI and ASI, and then there's a relatively small number of founder individuals around that time, and they expand, and there's almost no gene flow into them from neighboring groups for thousands of years, even though people are living right next to many other groups. So somehow this group has maintained its near complete separation from other groups for thousands of years, despite living right next to other groups. There's been some anthropological work which has argued that caste in India is really an invention of colonialism. It was not as strong, and maybe in some ways that's true, but at least in groups like this, in the Vaisya, these groups really have lived next to other groups and somehow remained, at least genetically, distinctive for thousands of years. You can see the impact of that in the genetic data. So to summarize, genetics documented a cultural change after 4,000 years ago. At the end of the Harappan civilization, there's a convulsive mixture to form the ANI and ASI, which then mixed and turned, and then there's a profound switch to endogamy. Now, my last slide. It creates a big medical opportunity in India. It's very clear that about 1/3 of people in India are from groups with clinically significant founder events. That is, a relatively small number of people, like in the Vaisya, who give rise to very, very many descendants today. So we further worked this up in a paper we published in 2017, where we surveyed almost a few hundred different groups all around India. There's thousands, but we surveyed close to 10% of the recognized groups. For each one, we measured how strong their founder event was. And if you show here in little red dots, groups in Europe with clinically significant founder events, such as Ashkenazi Jews or Finns, and these are groups that have known catalogs of rare recessive diseases that arise due to someone who was one of the founders happening to carry one copy of Tay-Sachs disease or something like this, and then they have lots of descendants, and when their descendants have a kid together, 15 generations later, that kid suffers from a severe disease. The stronger your founder event, the more such diseases you have. There's many known groups with founder events. There's Hutterites, there's Amish, there's people in Finland, there are Ashkenazi Jews, there's Sardinians in Europe. But 1/3 of the groups we saw in India had founder events stronger than these, including much stronger. And these groups include groups with millions of people. Including people like Baniyas with like millions of people, but with founder events 10 times stronger than in Ashkenazi Jews. And proportionally, one would expect many more such rare recessive diseases. Amazingly, people are not tracking these diseases. In groups like Sardinians or like in Ashkenazi Jews or Finns, you get when you go and you're about to have a kid, you have panels of genetic variants which are known to be risk factors for disease in these populations, and you could be tested for them. There's interventions, especially in communities where there's still arranged marriages. For example, amongst Ashkenazi Jews, people often don't even get introduced to each other if they are both carriers of a type of mutation. And in Orthodox Jewish communities where there's arranged marriage, some of these diseases just don't happen. Whether or not that's an advisable, a good approach, these are things to think about in a South Asian context. A rough estimate is that there's 100,000 babies born every year who are going to die in their first years due to one of these diseases, which could be completely avoided through these types of interventions. So I think I'll now move to Priya.
I think David's talk was a really wonderful introduction to what I will tell you about. We discussed, so in typical genetic studies, as you know, our genomes are 3.2 billion bases, and we have 2 copies because all of us are diploid. And in typical genetic studies, we look at selected sites in the genome. As David mentioned, we would look at like 600,000 positions in the genome. But now sequencing has actually become quite cheap, and we can, instead of just looking at 600,000 positions, we can actually look at the entire genome, so all 3.2 billion bases, and 2 copies of that, so 6.4 billion letters in the genome. Turns out majority of these bases are actually fixed between the 2 copies of your DNA. And so we tend to look at the sites which are typically variant. And we have now started looking at this in human genetics studies. There are now very large studies which look at thousands or hundreds of thousands of individuals. And this gives you really a lot of power to make very precise inferences about evolutionary history as well as different traits. And in a recent study, in collaboration with the All India Medical Institute in Delhi, as well as with colleagues from University of Southern California and University of Michigan, we generated data for 2,700 different individuals across India. We tried to sample all different states, people of different languages, people of different social status. And what we did that was different from what David showed is that rather than sort of doing group-specific collection, we actually did a grid sampling. So in every state, we tried to collect 200 to 400 people from each different state just to see how much variation there is, even if we don't get information specifically about the group. We also have a lot of ethno-linguistic information and health information. This is actually part of a large study called the Longitudinal Aging Study of India. And we also have health information, and we are trying to also add more information and diagnosis of Alzheimer's disease and dementia and other phenotypes in this data set. And when we look at this data, this is the same kind of plot that David showed, the principal components analysis, where we look at data from West Eurasians. I'm not sure why they didn't show up, but there's a large cluster of individuals over here in pink, and another cluster over here of East Asian individuals. And when we look at that, we recover the same Indian cline, which is modeled as a mixture of this ancestral South Indians and ancestral North Indians, the ANI and ASI that David talked about. We have also looked at the extended models with ancient DNA, and pretty much most of the individual people living in India today have ancestry from these three groups that David mentioned, the ancient Iranian-related populations, as well as the steppe pastoralist groups, and also some indigenous South Asian ancestry. So broadly, we recover similar patterns as we have seen earlier. And in our data, we don't have very detailed caste information, but what we can start looking at is how related two individuals are, or how related the two chromosomes in each person's genome are. And if you find they're very related, what you expect is there would be no differences between those two genomes, and that would lead to what we call a run of homozygosity, which is basically there's no differences, so they are just homozygous regions. And we can then, if this is because of very recent marriages like consanguinity, in particular, say when two first cousins marry or an uncle and niece marry, you tend to see a large increase in this. And what you see is that there are very large blocks of such regions in the genome because the two parents are very related to each other. So what we did is we tried to split up these runs of homozygosity of two sizes to see how much is related to consanguinity and how much of it is related to homozygosity. And then we compared it with other worldwide populations like East Asians and Europeans and Africans. And what we find is in most groups in India, there is a lot more there's much less diversity compared to other worldwide populations. And in particular, we see that this is more pronounced in the south of India, where consanguineous marriages tend to be common. But actually, if you even remove all the very large segments, which are because of consanguinity, you also find many of these darker color segments, which are largely because of the founder events that David mentioned. So even by doing a population survey, we similarly recovered the same pattern that had been seen across different groups. In some earlier work led by a postdoc in my group, we also tried to infer when these founder events happen. David gave you an example of one of these groups, so Vaishya, where he dated that the founder event was about 2,000 to 3,000 years ago. But we thought that maybe we'll look at a lot of different groups. And so here I'm showing you roughly 50 to 60 groups where we have independently dated when the founder event occurred in each group using similar ideas that we had used for looking at when the ANI-ASI mixed with each other. But this time, rather than looking at two diverse populations, we are looking at these segments of homozygosity or segments that are shared across individuals. And by doing that, what we find is actually these events occurred in the past 4,000 to 400 years. In many of these groups where the timing is very recent, what we find is that there is very strong and continuous evidence of founder events. And this is because these populations have very small population sizes. And so they had very few founders. And so the signals are even very strong. But most of this predates the colonial rule in India. So it's all from the past 400 to 4,000 years. And it's all postdates the ANI-ASI admixture. And there are some sort of similar patterns that are also, some of these models are also reflected in ancient Indian texts, where if you look at the Rig Veda, in its earlier descriptions, it describes a society that is very free-moving. It has chieftains from different communities. There's a lot of movement across groups. But in the later parts of the Rig Veda, the caste system is introduced. But at this point also, it's more about grouping of people of different professions or different occupations. But in the later parts of the Rig Veda, and in particular in the publication of the law code of Manu, what you start seeing is that there is descriptions of societies that are much more rigid, where there is forbidding of intermarriages across different caste groups. And similarly, we also see that the ANI-ASI mixture was bringing these very diverse people together, whereas once in the later parts, now we start seeing that there is no mixing across different groups. We can also, like David showed you, this plot of trying to quantify how small these founder populations were. We also extended a method from his lab to then try to do a similar analysis. And what we find is over here, the bars actually are how strong the founder events were. They are sort of anti-correlated to the number of founders you might have. So if you have a very long bar, the number of founders are very few. And in fact, the longest bar that you see is for the Onge population from Andaman Islands. And this group has more than 10 times stronger founder event than Ashkenazi Jews. And actually, the census size of this group is today 100 people. Another group that David highlighted was this Vaishya group. Now, Vaishya group is actually known in Andhra Pradesh, or in some communities in Andhra Pradesh, to have a mutation that leads to very severe response to anesthetic drugs. They tend to get muscle paralysis in response to anesthetic drugs. This is because they have a mutation in this gene BHCE, which responds to anesthetic drugs. Now, such a mutation, which has such a deleterious effect, is actually not seen anywhere else because selection sort of keeps these mutations out. If you look at other worldwide populations, none of the other worldwide populations have this mutation. This mutation is not seen in very large surveys of human populations outside of India, where 150,000 genomes have been sequenced. And you only find it in very few copies in South Asian populations, where here they had looked at over 4,000 people from India, and it's present at a 0.3% frequency in all of India. But actually, if you collect data from Vaishya, this is collaboration with Professor Thangaraj at CCMB. In this group, you find that such a deleterious mutation is present at more than 10% frequency in frequency. And it's sort of, this is a really good example of sort of the kind of idea that David ended his talk with, of how there's a major medical opportunity to try to identify such mutations and catalog them, and then potentially provide prenatal counseling or genetic tests. In this case, actually, there are many anesthetic drugs available on the market. And so if you have this ancestry, and if you know that you might have this mutation, you could get genetically tested. And then when you have a surgery and need anesthetic drugs, the doctor could just use another anesthetic drug, because other anesthetic drugs actually work on different pathways and they don't impact this. So there's a major potential to reduce disease burden by cataloging these variants. Now, another thing whole genome sequences actually allow you to do is that you can look at all the variation in your genome and then start understanding what is the source of this variation. And one super exciting discovery in this area has been the discovery of in understanding our relationship of modern humans with Neanderthals and Denisovans, something that in 2022, Svante Pääbo got a Nobel Prize for, and David's lab was very central centrally involved in the analysis. What we have learned, or a cartoon model of human evolution, is that modern humans arose in Africa about 200,000 to 400,000 years ago, and a group of them left Africa about 50,000 years ago. And as they left Africa in this journey, they met with Neanderthals and later met with Denisovans. And today, most non-Africans, or all non-Africans, have ancestry from Neanderthals, and most Oceanians and Asians have ancestry from Denisovans. But in these analyses, majority of the analyses have often focused on Europeans. More recently, there is also focused on East Asians, but there hasn't been a very systematic survey of archaic ancestry in South Asians. And so that's what we set out to do with our data. We tried to quantify how much Neanderthal and Denisovan ancestry is there. Again, think back to the plot that David showed of when ANI and ASI mix. Now, in this case, Neanderthals and modern humans mix with each other, but little bits of DNA from Neanderthals will persist in our genome over many, many generations later too. And we can try to identify these bits and quantify that. And we find on average, South Asians have about 1.5% Neanderthal ancestry and about 0.2% Denisovan ancestry. We can also compare this with other worldwide groups, and we find that we have very similar amounts of Neanderthal ancestry as Europeans or East Asians. East Asians tend to have a little bit more, about 20% more. And in terms of Denisovan ancestry, we also have similar amounts as East Asians, much more than Europeans, and Oceanians actually have much larger amounts of Denisovan ancestry, about 3%. Now, again, the other thing that you can do is you have these very diverse bits of DNA in your genome. We can start sticking them together to actually reconstruct an archaic genome from our own genome. So we can make Neanderthal genomes by just putting pieces of the Neanderthal ancestry that we find in our genomes. And what we find is that if you do that and take data from Europeans, this is one of the largest surveys of archaic ancestry in Europeans, you recover about 1 billion bases of the Neanderthal genome from European genomes. But because Indians are so diverse and they are not really one group, but actually a collection of many, many different groups, what you recover is almost 50% more of the Neanderthal genome out of the South Asian genomes, even with 1/10 of the sample size. And that's also true for the Denisovan genomes. And so studying Neanderthal and Denisovan genomes in South Asians has a major opportunity for understanding this evolutionary history. And this history is not just interesting from understanding our evolution, but there are also many functional traits that this may have impacted. In fact, we can look at where in our genomes and build sort of a map of where in our genomes there is Neanderthal or Denisovan ancestry. And if one of the largest hits we find is this one region on chromosome 3, where there is 50% Neanderthal ancestry in South Asians. And actually, during the pandemic in 2020, a paper from Swante Pebo's group was published highlighting how this region increases the risk of severe COVID and increases the risk of hospitalization by almost 40%. And in South Asians, this very deleterious variant is present at 50% frequency compared to Europeans over here, where it's present at 7% frequency. Again, maybe because of the founder events, it's drifted up to very high frequency. Yeah. Yeah, we have actually looked at that, and we see that it is not randomly distributed, but groups that have, groups in the South tend to have a little bit higher ancestry in this region. And similarly, we can look at the Denisovan ancestry. And in this case, we find a region on chromosome 6 where there is unusually high amount of Denisovan ancestry. There are many, many shared variants from Denisovans. And we find that this is related to immune function because in this region, there are many immune-related genes. And so just to give you sort of, a rough sort of summary of these different evolutionary events, or if we had to reconstruct the history of India, people in India have been living or in South Asia have been living there for the past 50,000 years. They all have ancestry from Denisovans and Neanderthals. Farming, as David mentioned, comes to India about 9,000 to 5,000 years ago of different types of crops in different regions. And then there is this mixture of the ancient Iranian populations with the indigenous Indian groups, which we have roughly tried dating by looking at these ancient samples that David mentioned from Goner, and we date that to about 7,000 to 5,000 years ago. And then the steppe ancestry comes about 3,500 years ago. And today, all our genomes are mosaics of these different little bits. We have, in some parts, Neanderthal ancestry, in some parts, ANI ancestry, in other parts, steppe ancestry. And then in the past 3,000 years, what we have seen is that there are very strong founder events. And so with that, I will just like to thank my collaborators, in particular, Dr. Day, who collected all the data from All India Medical School, and my postdocs who led this analysis. And I'll quickly switch to Vijay's talk.
This has been an amazing foundation that's been laid on the genetics of India and its understanding. I think both David and Priya alluded to sort of the health implications, and that's what I'd like to dwell on during my part of the presentation. And so I'd like to start with, this. Actually, when Hitesh and I talked about this session, I think we really wanted Abraham Verghese to be here, but unfortunately he's traveling. But the Covenant of Water, actually, let me step back and point out that the history of genetics begins not with Gregor Mendel's experiments with peas, but with people long ago noticing that family resemblances and vulnerabilities are so distinct that this could not be just because of shared environment, right? And that's where the focus of genetics actually begins. And I think Abraham has really sort of used this as the spine to tell a story, a multi-generational story of a Syrian Christian family, in Kerala, in Travancore, and it goes across 8 decades. And it turns out that there's a founder mutation of some kind in it's called neurofibromatosis type 2, NF2, which manifests as acoustic neuroma, or essentially tumors that go on the inner eardrum, which actually causes drownings. And so it's an extraordinary story, both of human genetics and, of course, a wonderful fictional tale as well. So when, and it's probably one of the few fictional books that both makes it to Oprah Winfrey's shortlist and gets reviewed in PLOS, right? So I think, it is, it is a story worth reading from that perspective as well, right? So I think the story also essentially points us towards the challenges of genomic health insights that we're starting to get in India. And I think both David and Priya alluded to these strong founder mutations because of the practice of endogamy and consanguinity in our population. So I actually became strongly aware of this back in 2013 when a group of us built India's first clinical genomics laboratory, which was actually, clinical grade. So we could do next generation sequencing, and we were CAP accredited. So clinicians could actually use the results from our lab as diagnostic quality, and we started seeing a lot of rare disease patients, right? And so in 2014, we actually started rallying a whole bunch of patient communities and stakeholders from the Indian Association of Medical Genetics, Dr. I.C. Verma from AIMS. We lost him recently. He was kind of one of the key figures in pushing work on rare diseases in India. The DBT secretary at the time, Dr. Vijay Raghavan, actually suggested to me not to use the name rare diseases because it kind of dilutes the public health sort of focus on this. So since then, I've been calling it orphan diseases, which is kind of what the Europeans also use as a descriptive term, of course. But orphan diseases can go beyond rare diseases, but it is a good way to talk about them. There have been sort of fits and starts towards a national policy, but I think by around 2021, we finally arrived at a stable national policy, and the policy actually it's quite detailed, and I wouldn't want to get into that. But I did want to point out that there's been an interesting social sort of dynamic of judicial activism, which has kind of pushed the creation of some of this policy. And I think, it's really worth studying from a social science perspective of how this judicial activism has interacted with the policy creation. When I started spending some time here at the School of Public Health, I think the thought was that it might be useful to start thinking about how to take this policy to action and actually think about a mission and towards that. And so I've just, this schematic is just my vision of how we would actually build something like a national mission or a master plan towards working to what I call no disease orphan by 2030. And therefore, when Tarun and others reached out to me about the Lancet Citizens Commission to think about universal healthcare by 2030, it sort of fit in some sense, my own personal mission into that framework of thinking about this from a mission perspective. I think some of the work on initial research work that has been focused on things like innovations in gene therapy and CRISPR editing, etc., I think have been strongly supported by ICMR and the Department of Biotechnology. And in some sense, since Soumya is here, I think some of the early thoughts around the guidelines, ethical guidelines for some of these research areas, I think were formulated by ICMR in those days. And in fact, one of the remarkable sort of N of 1 studies on antisense oligotherapies that was put together, was actually supported by ICMR, by Samya's team, was for a child with Duchenne muscular dystrophy. So I think there's been a lot of movement, and I think the question is, how do we actually get all these pieces to now coordinate and work towards no disease orphan, right? I'll focus mostly on what I call democratized genomics, and the reason to think that we are now at the end of diagnostic odysseys for many of these founder mutations or challenges with rare diseases. And so that gets into a little bit of technology and economics, but we'll sort of focus on that. Some early learnings that have come out of this democratization, and then a little touch on how we could build some social awareness about by leveraging things like digital technologies to communicate to society and create awareness. And on democratizing genomics, I think many people forget that there have been sort of two exponential laws that have really affected how we've progressed. And in really the last 50 years or so. And it's remarkable that information science, information technology, and molecular biology have really sort of had this overlapping timelines. And both have been driven by these exponential technology laws. And of course, we all understand Moore's law and what it's done for us, and how, by about 2006, when Moore's law entered the second half of the chessboard, so to speak, 2 to the 32 times, it created remarkable changes, right? Deep learning, or the concept that you could create something like an Aadhaar, was only possible because of, we had entered or we had passed a tipping point. So the question is, where does that tipping point come for genomics? And I think it's already come from $1 million for a gigabase of sequencing, which was what roughly 20 years ago it cost, to we are heading towards $1, right? And that's the scale at which this technology has improved. And today in India, we are able to do it at somewhere between $2.50 to $5, right? And I think $1 is certainly on the way with not just Illumina, but Ultima, Element, the Chinese MGI machines, and so forth. So I think this is what is going to democratize genomics in many ways. Commercial thinking by groups like Reliance have, of course, completely democratized telecommunications, for example, and the cost of being able to use mobile technologies, and their recognition that the tipping point had been reached with Moore's law is really been sort of a driving force for that democratization. And Mr. Mukesh Ambani actually reached out to us to tell us that he believed that genomics was ready for the same kind of democratization. And so for the last 2.5 years or so, we've been working with the Reliance Group to actually try to achieve this. So we're at a point now where for about $150, we can actually do very deep sequencing of at the whole exome level of an individual and also provide genetic counseling. So this is roughly around 12,000 rupees or so, right? And the hope is, of course, that this will be brought down to at least a third fairly soon, and maybe even less. Not very long from. We're not that far away from it. Now, what does all this start to tell us? Well, I think what we're starting to see from this deep sequencing is that, we're certainly getting insights on pharmacogenomics. I think Priya alluded to the Vaisyas who have a certain reaction to a certain type of drug. There are a lot of these pharmacogenomic markers that can be found, and it turns out that well over 70% of the Indian population that we are studying, and I just want to clarify that right now the data is based on hundreds of samples, but it's growing very rapidly. So we will soon have data on thousands, and that I believe the numbers are already stabilizing. So these might not be too far from what is actually the case, and I think the fact that Plavix or clopidogrel doesn't work for more than half the population is actually very significant again. I think David already alluded to the fact that maybe about a third of the population of India might have some types of founder mutations. And this is, again, starting to hold up. So we see about 85% of 41%, so which is roughly about a third of the population does have carrier risk of some kind or another, which are these founder mutations, which are carried. These are strong monogenic markers of carrier. These are mostly recessive. So these are in healthy populations, not in disease populations, right? And and then there's cardiovascular risk, there's cancer risk, and so forth. But I'm going to focus on the carrier risk. And for example, 1 in 25 Indians is a carrier for beta thalassemia, right? And which means that if we do nothing, about roughly 10,000 babies who are thal majors are born every year, right? And so the disease burden is now starting to strike home, and that we need to do something about it. Just as a personal note, I'm a carrier for spinal muscular atrophy. And I never found this out until we did this deep sequencing. I'd had my whole genome sequenced. I'd, of course, done all these 23andMe and so on. But copy number variants don't show up unless you really do deep sequencing. So I think it's an important fact here. So communication is, of course, important. And here's a little vignette. I think there's no volume, but that's okay. It's just to convey. This is a child suffering from thalassemia who's suggesting to her teacher that she and her partner get tested, before they go into planning a family. So, and then they realize that you can do it with just a simple blood test, which And they obviously raised the question, why isn't this done on a national scale, right? And the fact is now many states are implementing screening of at pregnancy of, particularly in the government hospitals. I alluded to the fact that there's growing capacity. I think today India is now capable of probably doing something like 300,000 whole genomes per year. And this is just Illumina. I think many other platforms are becoming available. And I'll sort of end here with this recently announced project that has reached its conclusion. We have 10,000 whole genomes that's been funded through the Department of Biotechnology to bring out the Genome India project. And the commitment given by Dr. Rajesh Gokhale, who's the Secretary of the Department of Biotechnology, is that soon after the publication, which is expected to be out by July or August of this year, this will be made into a public good. So the idea is that just like with things like Aadhaar and financial inclusion, we should be thinking about genomic inclusion as well by making a public digital infrastructure around in this India Genome Project. So this will be housed at the IBDC, the Biological Data Center, which is being built in the National Capital Region and should become available by the end of this year, is what we've been told. But I think again, I'd like to leave this slide up there because it might be useful for the discussion that follows. Right. Thank you.
So thank you, Vijay. I'm the chair of the session, so I'd like to start with a question myself and then open it up to the floor. So one thing that I've found kind of distressing is that in a context of Genome India is a major initiative. It's going to be 10,000 people, whole genome sequenced from diverse people around India. Project like Lasidad that Priya talked about is 2,700 whole genomes. But in context of what's going on in the world, what's happening in South Asia and India is actually, it's lagging far behind everywhere, even Africa. So in the UK, there's been already for many years, this UK biobank study with 500,000 people who have been whole genome scanned. With now everybody has whole genome sequences. It's 50 times more people than in India, but the country is much smaller in population size. The United States has this All of Us project with 1 million or so people. There are huge European projects. There's actually much more African genetic analysis and much more East Asian genetic analysis. So I'm wondering how we're going to get, what we need to do to get from where we are right now to having these biobank scale studies that in those contexts of those other populations have really it's been very clear that to obtain subtle understanding of risk factor for common disease as well as rare disease, really 10,000 samples, 2,000 samples is really not going to yield the same insights that hundreds of thousands of samples or millions of samples will.
So I have an easy answer. I think I'm looking at Tarun and saying, can the Mittal Institute actually drive a focus on genomics in South Asia as a major initiative, right?
So David will remember that I initiated a conversation with Vijay Raghavan, and I think we had a meeting with Eric Lander at the Broad, and we sent you to the Jaipur Lit Fest and beat a lot of drums, and then we hit a brick wall.
Yeah. So I think part of the challenge
God knows.
Yeah, no, I can tell you what the brick wall is. I think the brick wall is that this, sending samples out, right? And does, and I think the difference now is because of the pandemic. I think we've now built enough infrastructure in India that the samples don't have to leave the country. And we can still have advisory roles that some of the thought leaders here can play, but the samples don't have to leave the country. They can be sequenced there. The data analytics, India is already, a powerhouse at. So there's really no problem now to actually propose a project. I think back in even 5 years ago, this was a challenge.
I think the other challenge is most South Asian countries take the privacy issue very seriously. So how you convince any government in the region to share any data is
Right.
And it's for real reasons.
I mean, another challenge is also the cost for this. So in UK, the UK Biobank sequencing has been done in partnership with companies like Regeneron Genetics has effectively funded this. And there may be sensitivities of, bringing in companies, because even if it costs only $100, that's a lot of money in rupees, and that's a lot of money as you scale to 500,000 people or 1 billion people if you really want good representation.
But certainly, I think 100,000 is within reach. I think is of the budgets today, right?
We're doing questions, and I think Sheila also had a question, so I'll bring it over to her next.
Thank you. So on the slide where it says that genomic data will be available for public access, is that internationally? Like, will researchers here have access to that data?
Yeah, good question. I really don't have the answer to that. I think in India, when research agencies essentially say that data is available, there's a process by which you apply and you're qualified. I think usually a genuine research project will be given access. But, whether if there needs to be an Indian institution as a partner is, those would be finer details that would be worked out as a policy by the IBDC. Right.
Okay, thank you.
Can I make
one follow-up question?
I just wanted, we can have a follow-up question also, but I just wanted to make a quick comment. The UK Biobank example is kind of an amazing example. So they were quite early on thinking about creating this resource, and then they ran the project and continue to run this project in this really quite extraordinary way where the data are made available very internationally. It's not posted online, but qualified researchers, after signing confidentiality forms of not reposting the data and so on, are able to analyze the data. And they haven't privileged internal groups particularly, and so they've shared the data simultaneously with all researchers. And the result has been this absolutely massive amount of research, thousands, tens of thousands of papers on this data set, incredibly valuable insights about the history of that particular population, which has served the country incredibly well. And so, it's an interesting model, and it's not obvious that one loses by thinking very carefully about that and inviting people to add value to that resource, whether they're local, which I'm sure would be the greatest added value, but also internationally.
Yeah,
What's your,
okay,
go.
Yeah, just to add to that, if privacy is a concern, which may be heightened in South Asian and other communities, what UK Biobank has started doing with whole genome sequences is that you actually can only analyze the data by logging into their server.
Yes, right.
You cannot download the data, you cannot make copies of it so that there is no risk for distribution. There's no risk of, if they share the data with you and your computer gets hacked. And that, ensures the privacy. And it's actually, I completely agree with David, it's been an immense resource for all scientists. And we have learned a lot about complex traits, about the history of the population.
And I believe that, if you want research to happen that's focused on South Asia, inviting people in to analyze the data could be an interesting model to think about. What's your other question?
I was just going to ask what type of phenotypic data is linked to the genomic data.
I'm sorry, I didn't.
Phenotype.
Oh, to the genomic data that I presented?
In Genome India, like what?
In Genome India, right. I think the phenotypic data, I mean, in many ways, the Lassi instrument became kind of the default instrument that now is being used for all these large genome projects. So, the longitudinal aging study of India, which was actually spearheaded by Professor David Bloom at the School of Public Health here, had developed an open instrument, and that has been adopted by the Center for Brain Research, which is one of the nodal agencies of the Genome India project, and is being used or adapted for several of these genome projects in India. So the Lassi instrument is actually a phenomenal phenotype, and I guess was used for the Lassi DAD as well, right?
2,700 genomes that I showed.
Right. That's right.
I have a question.
I think the mic's here. Yeah.
Yeah, I have a question that in a way ties the first panel to this panel. So the first panel talked about India specificity and the fact that the large language models are developed for English, and how to get over those problems in a country with huge linguistic variation. So David, one of the interesting, but there are two interesting findings from your research that have been talked about a lot. One is the point about India not being one huge population, but many small populations. And the other is nevertheless a correlation between genetics and caste structure, which has been picked up and understood in a variety of different ways. So I'm struck at your presentation, Vijay, about how the terminology remains the terminology of the North. Even the traits that are being talked about are the traits that are widely studied already. I mean, beta thalassemia first came into prominence as a Mediterranean area disease and not specifically India. I was interested. I don't think I saw sickle cell in your chart, right. but that's another one. But what is the correlation between the small populations and the rare/orphan diseases, and then broadening out to the socioeconomic side of things? To what extent are the kinds of genetic quote defects unquote that are being focused upon correlated with the more ancestral northern versus ancestral southern, or the mixing of populations? I mean, we're talking about democratizing as if we're on already a flat playing field and anybody can have access to anything. But of course, that's not right. And so I'd be interested in the sort of socioeconomic stratification along with the genetic stratification in the problems that you're discussing.
No, absolutely. And I think, there's sort of two issues there, but let me start with what I see as the more important one. So, for example, soon after Abraham's book came out, there was a condition that was reported in the Indian press. In 3 villages just outside Mysore in Chamarajanagar, there seemed to be some kind of a covenant of light. So this was a kind of a retinitis, genetic disorder trait, which actually, when children who have this trait are exposed to light, bright light, actually both have loss of sight and sort of degeneration of health, and many of them die quite young. And the villagers really believed that this was a curse. And so a medical geneticist from JSS Medical College, which is linked with Mayo Clinic, actually went to these villages and has begun a process now of and has found philanthropic sources to actually do sequencing and start the genetic counseling in these villages. So these are small examples, but I think it's starting to go deeper and to that democratized levels. The kind of vignette that I showed, for example, will leverage the Vani project that Manish Gupta talked about, and we will communicate these as little chat mobile clips like Armand does with Milind, and communicate them in dialects. So the Vani project, which is now at the Indian Institute of Science, using these speech patterns, will be able to actually put out these chatbots, which can actually speak in a very local dialect. And we're not that far away from being able to implement many of these. Yeah.
I think that's I'm going to answer that question along a different axis a little bit. I think that one of the things that I've found a little bit frustrating in the discussion about medical needs that are genetically related in India is that there seems to be a disconnect between talking about conditions that are common across India and ones that are specific to particular groups. So there is a well of unique variation that's present in South Asia that's not present in very many other places outside of South Asia, that's heavily represented in the ASI, for example, but also to some extent the ANI, but especially in the ASI. And that tens of thousands of years of separate population history has accumulated mutations that are more common in that population and might perhaps be related to increased risk for hypertrophic cardiomyopathy or lipid or metabolic or diabetes-related disorders and so on. And so people tend to focus on these India-wide phenomena that are more common in India epidemiologically than elsewhere. And that's a different issue, looking for the genetic variants that are more common in the ASI than in other population, than identifying these orphan or rare diseases that collectively are individually extremely rare, but put together are extremely common. So I think there's these two separate issues about rare diseases and the attention to those, which needs to be disease by disease and group by group, because they're going to be completely random and unsystematic in what systems they affect, and the others that are broad and common and widespread and affect large numbers of people. The other thing is that there's environmental and cultural differences that interact with the genetics, or don't even interact with the genetics particularly, that are not genetic at all, that are specifically affect South Asia more than other places, and should not be really thought of necessarily as a genetic phenomena that require attention. So people with any genetic background in a South Asian cultural or climatological sort of environment would be susceptible to. So I think attention needs to pay to all these axes, the particular diseases and phenotypes that are common in that area, the unique well of South Asian variation that's common across people, and then the very specific ones. And I think they need to be thought about sort of clearly and independently. I don't know if that was a partial answer to what you're saying.
Yeah, and I would just like to add one more point. This is kind of, you alluded to this point that maybe there is potential for discrimination because of identifying these variants that are present in some groups but not others. And that actually, I think it is a real serious concern, but I think we can look to the West where there are many, many founder groups like Ashkenazi Jews, Hutterites, Sardinians, French Canadians, and they have also identified these mutations related to disease variants in their groups. And those catalogs have been immensely beneficial for reducing disease burden in these groups. And we, as Indians, could take benefit of that. There are so many children that die of these very deleterious diseases in early childhood. And these are, just by screening or looking at the Vijay example of reducing, the solution there is very simple. There's no need for even concern. You can use another anesthetic drug. And by cataloging these, we can really leverage the power of the science.
So we have one more question. It's just to sort of emphasize what Priya said. It's trivial to identify a panel of risk factors for disease in these founder mutations. So if you were going to try to identify the risk factors in Sardinians or Ashkenazi Jews or Finns today, all you do is you sequence 500 people. It's very cheap. You identify all their rare recessive mutations that are predicted to be recessive, and you find everything that's known. So like with current technology, all the tens of millions, hundreds of millions of dollars that were put into sort of finding the rare diseases in Finns, you sequence 500 people, you run an algorithm, we would find them all. So it's like a very simple thing to do at this point using current technology. There's one last question up here.
Talking about thalassemia, India has, I think, about 10 to 15,000 babies born every year. So what kind of diagnostic tools are present in rural India, where there's probably not much awareness? And what is being done currently in India? Is the government doing anything about it, or how is that being tackled?
Yeah. So I think, these are globinopathies. So both sickle cell and thalassemia are looked at because it's the same gene. And there are screening and, confirmatory tests. The government typically spends something like 100 rupees for the screening test, and I think is willing to go up to about 500 rupees on the confirmatory tests. And these programs now are fairly widely implemented at least in Odisha and Gujarat. And in Tamil Nadu, for example, on the bleeding disorders, there's a lot of work. So hematological disorders have actually been looked at very carefully, and both from a public health perspective and from a perspective of treatment, I think there's, and sickle cell, of course, as Sheila pointed out, is a very large problem in our tribal areas. And the government is at least now very seriously pushing for universal screening for being able to detect sickle cell disease. There are some ethical challenges of ID cards and so on, which are being discussed, but I think there is at least an attempt to start thinking about this as a public health challenge. Right? Yeah.
I was just going to suggest that we should conclude the session.
Right.
No, what did you want to say? Yeah.
So I just wanted to have a response to David and Chandru's initial response to that. I do think we're at a different moment, right, than, I don't know when we did that, 8-9 years ago, and then even a few years ago. And I wanted to say that there is a level of confidence in India, maybe swagger, not wholly justified, but partially justified. But I wonder if there's a way to use that confidence, because the dual of confidence is insecurity. And I think it's the insecurity that blocked the previous effort in the end, is that I don't really know what you guys are up to, so I'm going to stop it. And that's what I wanted to observe, in addition to the evidence that the Digital Stack Public Goods Project has begun to be recognized as a technological achievement, including in Japan and France and other places. It's not just the poorer countries. So this is feeding back into what's into the political economy, and I wonder if it's a moment. And then if you've got, people like Mukesh Bhai, Ambani's, and so on, putting their considerable influence behind it. Sometimes good, sometimes less so, but in this case, potentially for the good. That's another sort of complementary factor that might make it. It's a judgment call as to whether the best time is today or 2 years from now or whatever. But it is different. So I echo that.
It's just obvious to me that the elements have come together to make it reasonable to plan an ambitious biobank scale studies in India. There's many reasons for this. One is that, as Vijay said, the cost is very small. It's sort of similar to the cost of a complete blood count at this point, which is a routine medical sort of analysis that happens to sequence a person's whole genome or to collect an equivalent amount of information. And secondly, it's a commodity now, sequencing. It can be done anywhere. You don't have to send it to, out of the country. You can do it into the country. You can send it out of the country. It's just a commodity. And thirdly, there's well-organized groups of scientists who can help to support such an activity. And fourth, the added value of doing this in India is huge, more than in most other places. So if you're going to do another biobank in Italy, the added value of that's going to be relatively small compared to the existing biobanks elsewhere in Europe. But the biobank that you build in India is going to be a greater added value because of the greater distinctiveness of the population ancestrally, because of the greater diversity in terms of subgroups, which means that the findings from it can be that much more impactful and less generalizable from existing other biobanks. So it just obviously should be done. It's not expensive, compared to many other expenses, and it's just obvious that it will pay fruit and it will be useful scientifically, it will be useful research-wise, it will be useful as a resource. And with careful thinking, it could be done in a way that's non-threatening, safe, that alleviates the security concerns that people might have and appropriately have.
Can I suggest that we take a no more than 5-minute break, I'm told? Yeah.
We take 5 minutes.
But please join me in thanking the panelists.